So I would just like to know from you all what you think are the major challenges working with the mass spec. So we have seen many applications, areas. So what is the top, maybe one challenge each one of you can just list down, and it should not be the repeat one. So we'll start with you, Vaishali, from this side.
One of the major challenges having trained folks, really trained scientists, who not only knows how to run a mass spec or do sample preparation, but understands the data. And basically, mass spec is such that you need to sit in front of the computer and really, no matter how sophisticated protein metrics, how sophisticated the software is, I'm seeing someone smiling, so he knows the pain of analyzing data. We do need scientists with a focus and with the trained mind and trained mind and eyes to understand the data, because there are a lot of false positives that can come out.
I think, Vaishali, you're right. That's one of the things we hear a lot is, getting people that, can run this, particularly as you try to move it into QC. I think I'll touch on something else, which I think is consistency across, across labs, across organizations. So, tech transfer within your own, your own sites, as well as if you think about from a regulatory perspective, can we compare apples to apples, or are we really looking at methods that are giving us very different information? And it's hard to kind of set a baseline for what, quality should look like. So I think that's the other challenge that we often see.
Thank you. I was going to start from consistency, but you took it. So the other challenge which I feel is there is about, when you're talking about proteins and peptides, their differential ionizability, and therefore how to quantify them in different samples, although the matrix and everything could be the same, but batch to batch. If you look at from a, say, size exclusion or cation exchange, where your reproducibility or precision accuracy is much higher, how do you achieve the same in the case of mass spectrometry?
Yeah, so I might have mentioned this before, and I think Vaishali also mentioned. So data analysis is a big challenge, and especially for high resolution mass spec. The softwares that are currently available, they have severe limitations, and the user's time, there's a lot of user time. There's noise removal takes time, redundancy in the data, and it's like so a lot of people ask if the mass spec hardware has advanced so much, haven't the softwares advanced? So it's the same story as satellites. So there are really high-tech satellites present, but there are hundreds of startups that are now analyzing the data that so somebody has made fancy satellites, but there isn't enough software. The other challenge that we feel is the libraries. So high resolution mass spec, the annotations are primarily based on libraries. So again, libraries are either not public domain or they are not adequate.
Thank you all. So I'm sure, with metabolomics, you can say the libraries is a big challenge, especially if you're analyzing the untargeted data. For antibodies, still there are ways to define. Antibodies are like the well-known molecules nowadays. So there are plenty of challenges, but despite that, I know all of you, we all have been using mass spec, and there are multiple advantages. It does give answers what we are looking for, right? So with that, I would just open up it for any questions from the audience. Anyone would like to? I can see many hands, but I'll go with Abita first. I saw her first, so let's start from her.
Thank you so much, Annu ma'am, and thank you all the speakers for the insightful session. So my question is related to new pick detection. So can somebody provide any insights related to validation, or we can say qualification of new pick detection, because it's one of the most challenging part of MAM. Thank you.
Maybe Diane, you would like to take it up?
I was going to say maybe Vaishali was thinking. Yeah, I think new peak detection is a challenge, and I think it's actually the challenge is really where do you set your threshold? I don't know that I have a good answer for that. I think there is a good section on this in the chapter, and I don't remember all the details. details off the top of my head. But I think that that is the key. Where do you set your criteria? And so I think going through some of the exercises and some of the experiments that help you figure out what is a true positive versus what is a false positive is really important in defining those criteria. You want to add to that?
Yeah, great. I mean, Diane kind of nailed it, but I do want to add that, yeah, as Diane mentioned, a structured workflow is very important. A new peak, I mean, is it an artifact of your peptide digestion, or is it there really? Do some post-degradation experiments in the initial stage to understand whether the new peak is consistently there. And if it's consistently there, then yeah, obviously you have to monitor it. And as you said, qualification, there are ICH guidelines around qualifications, right? And for mass spec qualifications, again, repeatability, LOD, LOQ of relative quantitation, those are becoming quite well known now. And you will find a lot of papers around that. But first make sure that your new peak is actually real and not an artifact.
So, just a question to you. In terms of new peak, are you talking about between the biosimilar and the reference product? Or it's just between and between the two? Or within a batch, within a product?
Within a batch itself.
So there are also, it's not that if you're looking at, say, the same manufacturing process and you're looking for a new peak, you will suddenly have to look at retention samples, older ones, pick them up, right, and also look at those ones, and verify whether that's actually new or not. Most often, you would have to go back to your clinical, if you're as a sponsor, right, you have to go for your clinical batches also and your tox batches. Look at them, and sometimes change of enzymes, change of columns can bring this about. You will have a better resolution. So then it's not a new peak. So that's how you have to go.
Okay, thank you.
That's a good question.
So good afternoon, sir. This is Manimaru from Waters. So again, my questions on the library. So for the targeted and untargeted analysis. So you told that you have more than 300 compounds that identified. So can you talk about that, how you identified the identification of 300 plus? Do you have any library built? So, and a little view on the unknown compounds, unknown metabolites, you have identified anything, and how you are confirm their identity using this HRMS.
Right.
Yeah, thank you, sir.
So if I understand how are the metabolites identified in the absence of standards, right? So if you have standards, it's a little bit easier. But in the absence of standards, one is matching of mass fragmentation pattern
Yes.
with the libraries that are available. Now, public domain libraries are somewhat limited.
Yes, very limited.
In two ways.
Yes.
One is the number of metabolites for which libraries are available.
Yes.
And number two, the quality of data.
Yes.
So quality meaning there are either fewer peaks in
Yes.
the mass fragmentation pattern, or there is noise in the mass fragmentation pattern. So when if you are familiar with the MS2 matching, so upside you get
Yes.
the sample and downside you get,
Yeah.
and how many of them are matching.
Yes.
So and if there is noise in both the library and the sample,
Yes.
or the experiment, then there is a lot of false positive that comes.
Yes.
And that is where the user needs to look at all of them and see based on their experience.
Yes. Especially it becomes more complex when you do with spin media, because it has a lot of other junks and other
Correct.
salts and other.
That is
compounds,
one.
yes.
The second approach that a lot of experts take is a diagnostic fragment.
Okay.
So, for example, carnitine, we do quite a bit of carnitines. So, 85 Dalton is a diagnostic fragment for carnitines,
Yes.
and so on. So, for diagnostic fragments, the limit must be present.
Yes.
So, if you give some annotation and say it's let's say acetyl carnitine,
Yes.
and in positive ionization, and the fragment of 85 is not present, then most
Then-
experts will not accept it as a carnitine. But
Yes.
the software might provide you as a carnitine because the software is based on something else.
Yes.
So it's some number matching.
So how about any unknown compounds, any unknown metabolites you identified, and is there a way you have any database built or any your software what you develop that's cross-talking with the public domain, like ChemSped or something?
Unknown meaning something which is not present in the library.
Yes, not present in the library.
Right, not present in the library, so there are again two approaches. One is to have a guesswork as to what it could be.
Yes.
You order the standard
Yes.
or possible standards.
Yes.
The option is to go for in silico fragmentation pattern.
Okay.
And then try to again come to some kind of
Okay.
possible annotation.
Okay, sir. Thanks.
So, Pradeep, I think, to this table now, I know there were 3-4 hands, and Nitin has been trying to ask questions, Nitin and Pradeep. So,
Yeah, so this is about the question is on the discussion that you did, Dr. Vaishali, but it's an open question. We can discuss on it. The basic question is how far we should go in supporting the early stage development using mass spectrometry. Why I asked this question, I'll give you a specific example. For say, you talked about clone selections and use of mass spec. Yes, totally agree. We also use mass spec in the process development per se, totally agree. But then there are aspects of product development, like you do a product intermediate whole time assessment. You do some label claim studies, which include freeze thaws, which include photostabilities, which include in-use stabilities. So how far deep do you assess the mass spectrometry based impurity characterization set limits, because the conclusion or the observations will change, and our conclusions on the stability of these intermediates or the freeze thaw cycles may change based on this. So what is your thoughts on that?
It's a great question, and I don't know whether I'll be able to do justice in 2 minutes or but I think how far means, I would say it depends on your capability of the company, okay? It is very, as you rightly pointed out, you need to have resources, you need to have high-end mass specs to analyze this. However, the mass specs is not going to replace your normal workflows of basic, as you said, post-degradation studies, and there are several whole-time studies. What it is going to do is it is going to help you make informed decision. So suppose you're doing clone selection. These studies are part of, your life cycle of a drug once you select a clone, right? And clone selection is based on several other parameters other than mass spec. Can mass spec act as an orthogonal tool and maybe give you, give you an answer which helps you make that decision in a more confident and an informed manner? That's where I think process development mass spec is coming from. It's an orthogonal technique that gives you a very powerful data, such as clone selection, such as sequence variants. For example, spent media analysis, where my colleague was showing how amino acid depletion is happening. If you can correlate that data with the sequence variant going up, think about it. I mean, that's where your data package to your IND submission becomes much more powerful. That's where it is.
Because for product claim studies, the basic understanding is it a regulatory expectations to cover mass spectrometry assessment, set acceptance criteria, and then define your stabilities for holds or all other stability claims that we make.
Mass spec is still going to be in a characterization space.
And
really,
just because...
but if you can support your data with your mass spec data, saying that, okay, you have optimized your process with sequence variant analysis, that makes your data powerful, more package more powerful. But regulatory agencies are never expecting that has to be done. But it just makes your package or story more powerful.
Just because you mentioned spent media, from process point of view in biotherapeutic development, we see that the basal media, the lot-to-lot variability in media's feeds that we use, that can lead to quality attribute changes or variabilities in batch to batch in terms of the product quality that we define. So what is your thought process, although it's not a mandate, we have not heard about this anywhere, of using the mass spectrometry for spent media, more than spent media analysis, using the mass spectrometry for doing a raw material release of the medias, of the feeds, because generally these are not disclosed. So we don't get a COA with all these values. So, and we do see variabilities due to this. So do you think this approach should be taken, or do you think it is something where the industry will move towards?
I don't see mass spec will move. I mean,
not mass spec particularly, but evaluating the spent composition of the metabolites in the media lot to lot that we keep
I
getting.
mean, as more and more information is coming up, probably we are heading towards that. But I don't see mass spec being the tool for that, as you said, because release testing again, um, the good thing about mass spec is it gives a lot of information, and the bad thing about mass spec is also it gives a lot of information. So what do you do with all the data that's coming up? So release testing has to be simple, right? So that's where, not probably mass spec, but obviously, yes, more orthogonal tools would come up. And I think my fellow colleague would answer that better than me.
Yeah. So obviously, during the development stage, there needs to be some development on correlating the media composition with the outcome, because the outcome is what is of interest, not the media composition. So those correlations, have to be developed. A lot of work will be required, and I don't know if mass spec will become a tool to for quality check of the media, but it might. I mean, because it can measure so many things, and currently in I don't know about the biopharma, but in general in the biotech industry, the quality control for the media is based on growth promotion test, and we know that it is not adequate at all, and anything will grow, even on bread, right? So yeah, right.
I think while you're right, in the Indian biopharma space, it's very common to buy media and then use it. But there are companies who are formulating their own media also, right? And they are using components on their own, keeping control on those components and formulating it. So they know what they're adding and how much they're adding. For companies in the Indian biopharma space or others who cannot do this, probably doing it as an RM analysis is a good idea, to know in future if when you do a scale up or something, if there's any change. It's not because of the media components, but something in the scale up, and that, therefore you can rectify it.
So next
Whichever technique
question.
you can use.
Yeah, hi. So I have a couple of questions pertaining to the SCP standards. So as you mentioned that you have come up with the...
Nitin, can you be little louder, please? So I think we have 3 here.
Am I audible now? Yeah, now it's audible. Huh. So as you mentioned that the USP has come up with 10 different standards basically. I would like to understand what is the rationale and what basis these 10 SCP standards have been selected. So why I am asking this, because there are certain SCPs which are known to be immunogenic, or we call it as a high risk SCPs. So that's the first question that I would like to understand from you. Second question is that what's the target number that you are targeting? I mean, like now so far you have come up with the 10 basically, so you have some target in your mind?
That's a great question. So, as we looked at this space, we worked closely with a couple of groups and organizations. One is we leveraged some of the HCP experts that were on our committee that originally wrote the 1132 chapter, and got their input on what did they see as the problematic HCPs, because that's really where we wanted to focus. The other organization that's really looked at this in great depth is, I don't know if you're familiar with Bioforum, but they have published several white papers that I think are publicly available in terms of what they had a working group around HCPs of what was considered to be high risk. So we worked with them actually before that white paper was out, did some surveys to really understand what were people most concerned about. So, I mean, we could make the list very long. We started really with the first 10, the first, we'll probably release two of those targets this year, and then it'll kind of continue from there. So I think we can continue to build as long as it is of value, right? It's at some point there's going to be diminishing returns, right? It may not make sense if nobody's using it, but this is where we'll look to you to tell us what else do you need as we get those rolling. So
Thank you.
So one last question, please.
Hi, I'm Mukul. I'm from Syngene. I have a question related to this SCP standard actually. So the purpose of SCP is like to identify the high risk, the immunogenic SCPs, and the SCPs which may cause some degradation to the product in terms of directly or indirectly, like linkages, creating this polysulfate and all these things, and directly some proteases are also there. So I have a small suggestion for this kit particularly. I mean, if you can categorize like one set for, let's say, for immunogenic model, and another set for the the proteases or the lipases model. So that will be really helpful for us. I mean, those standards are really very costly. If I go to market to manufacture individually, these are very costly. So one common set will be helpful for everything.
That's good feedback. Okay. Interesting to think about, right?
Okay, only one. I think I just want to appreciate Professor for developing such a good tool. So my question is, what is the end? Kind of like eventually, when we do everything, is for more biosimilar product, or I can say fingerprint biosimilar product, and as well as a good yield. So these are the two targets, apart from many other targets. So what is your next one? Because really, it's a great, journey, I should say.
Yeah. Thank you. So, so obviously this is work in progress. A lot more needs to be done in this direction.
My question is like any correlation has been done on these two aspects.
Correct. Okay. So we on our own we have done for E. coli. So there are markers in the spent media that correlate quite well with productivity or lack of productivity, both ways. So with E. coli that has been done, and now the ongoing work is on choke cultures to understand these productivity markers, so that you will be able to see if something starts accumulating on day 2 or day 3, you already know that things are not going well for that particular batch.
So last question for Diane. So sorry. So my question here is, once again, where it will going to end up? Because see, it's the double work for us from the analytical perspective, because we need to do it mandatorily with our classical ELISA method. At the same time, you are introducing the standard. So I mean, it's very difficult to replace everything, but in the halfway. So what is the thought process? I mean, we understand that we need to have this method. It's like, progressive part, but what is the end? Like, what is the vision? I mean, from the USP perspective.
And can you, I think I missed the beginning, were you relating to HCP or to MAM?
HCP.
HCP. Well, I think that this is, it's an orthogonal technique and it's part of risk assessment. I don't think that, today anybody is using mass spec as a release assay. It's really done in that characterization phase, and where I see it really being of value is it allows, during process development, people to, adjust their process so those high-risk HCPs don't come through. So I think that's really where we see it being used to inform, process development and inform characterization.
Sorry, if I...
The expectation of agency also will become...
Obviously, that's the question. We have to do two. I mean, I have to mandatorily do my ELISA method to say that this is my ppm limit. At the same time, it is always an expectation when it comes into the USP. So, agency will ask.
If I'm not wrong, recently there was a BLA approved where the HCP method is mass spec.
Yeah,
it's not ELISA.
Absolutely. No.
Right, so
I mean, like,
so
we are progressing towards that, but it's not the end.
They don't have ELISA as their HCP release method. It's only mass spectrometry.
That's highly debatable point, but we can...
That's what the BLA approval is.
In the interest of time, I would just like, I know this is a really interesting discussion, and now actually it's warming up, and this is a very debatable topic, ELISA versus HCP. We can spend hours discussing that. So I would just say we can have that conversation during coffee break, or after they all are here, so we can just catch up offline and then have those discussions.
I wanted to ask Professor Wangikar one question, if you allow. Just for the benefit of everyone else. So, what you presented, and most often what the work people do is a metabolite is limiting, and therefore
because
you
it's
add that,
already
it will increase the productivity. Any insights on, there is a metabolite which is in excess, and probably that is causing productivity loss, or it's inhibitory, there are some of those inhibitory. And in that case, what should be done?
Yeah, so again, this is work in progress. So we have been optimizing the process based on the data that I showed. and the conventional wisdom is that anything that is getting taken up quickly, you feed more of that. So it works only up to a certain extent and only for some of the metabolites. And it gave the productivity higher productivity that I showed for one batch, and for that one batch we have 3 other batches with slightly different feed combination which give lower than the control productivity. So we still don't know which is the right one, but we are getting there to figure out what are the key components that should be added.
So with that, thank you. So there is a small facilitation for the panelists. Thank you everyone for the wonderful discussion. We really enjoyed it, and to be continued outside. Yeah.